Good morning, one and all. Myself, Dr. K. Swati Priya, co-convener of the FTP Faculty Development Program on the theme Challenges and Innovations in Advanced Pharmacognosy. I welcome you all to the fifth day of the Faculty Development Program. Now, I request Dr. Jayalakshmi ma'am to introduce our today's resource person.
Thank you, ma'am. Good morning to all. Myself, Dr. V. Jayalakshmi, working as Assistant Professor in Srinivasarao College of Pharmacy. It's my immense pleasure to introduce you and welcome Dr. Rahul Kaushik, sir, for fifth
Thank
day
you.
of 1 week FDP on challenges and innovations in advanced pharmacognosy, organized by Cognosy Department of Pharmacy under management of Alvadas Group of institutions. Currently, Dr. Rahul Kaushik sir is working with Metro College of Health Sciences and Research Pharmacy, Greater Noida as professor. Sir did his BPharm and MPharm Pharmacognosy and PhD from Dr. APJ Abdul Kalam Technical University, Lucknow, India. Sir has 13 years of experience in industrial research and academics, and he has guided more than 50 undergraduates and 10 postgraduates. He has participated in various FDPs organized by government and semi-government institutions. He has authored more than 50 research and review articles in various international and national journals and also attended more than 29 national and international conferences and seminars. He has contributed book chapters in various national and international publishers like Springer's Germany, Springer Singapore, and Nova Publishers USA. He also serving as scientific reviewer for various publishers like Dover Press, Bentham Publishers, etc. And also he has a brand ambassador for Bentham Publishers. His area of interest are exploring herbs and Ayurvedic formulations acting on central nervous system with special interest in anti-epileptics and also pharmaceutical journalism, serving as educational board member in journals like Acta Pharmacon, International Journal of Pharmaceutical Education and Research. Sir has membership in various esteemed national and international organizations like Association of Pharmacy Teachers in India, lifetime member of International Natural Product Sciences Task Force, FHER, IPES, Prizal, etc. Sir, it's an honor to welcome you, sir, for today's
Thank you
session.
so much.
Now I request our resource person, Dr. Rahul Kaushik, sir. Thank you.
Thank you so much, ma'am. It's my privilege uh, to be here to present my research, my topic on the current trends in herbal drug research. First of all, I am very much thankful to the organizers of this uh, 1 week faculty development program. And I am very much thankful to the SRCP for giving me this opportunity to showcase some latest trends which is a need of time in herbal drug research. Actually, this presentation which I am going to deliver today, it is mainly focused on some aspects that we generally neglect in pharmacognosy, and I have given more stress on toxicological evaluation of drug or herbal drug. Moreover, I have confined my presentation on various new techniques which are being used throughout the world for toxicological evaluations. So, moving on to the first slide. Ma'am, please change the slide. Next. Okay. As you can see, that as per the statistics of World Health Organization, around 80% of the world population, they rely on herbs or herbal derived products for their therapeutic virtues. And we have seen an exponential increase in the demand for these herbal remedies in the last decade. And after COVID, there is an phenomenal increase in the demand for these herbal medicines. But you can see when the demand for anything is increased, then we also suffer some malpractices also. So this huge demand, it causes escalation in the rate of fraudulent activities like substitution and adulteration of the herbal materials and addition of synthetic molecules like generally we see in the muscle building powder. They usually claim it to be herbal, but they use steroids to show the efficacy of their product and to gain the false public interest in their product. So what we are going to focus here, uh, there is a strict need for quality products in the market, and for that we have to develop various standardization quality control parameters for authentication and standardization of or you can say the quality control of our products. So if we see, there are traditionally the parameters which we are using for quality control of drug. We can see there are various parameters in the next slide. But with respect to the safety concern, because most of our products which are being exported from India to the foreign countries, they are being sold in the form of as a food supplement, not as a medicament or not as a drug. And this is because we are not capable to supply good quality finished product because of substandard quality of our raw material and the lack of interest of the companies, the business tycoons in quality control of the drugs. So to address this aspect, an attentive toxicological assessment is the prime requirement which can eliminate the potential safety concern. So next slide, please. So these are the traditional pharmacopoeia quality control parameters which are prescribed by pharmacopoeias like Ayurvedic pharmacopoeia as well as our WHO guidelines. So these are the traditional things which we are going, and few improvements are there. New technologies are there, like HPTLC fingerprinting, DNA fingerprinting. And these are the need of time which we have to focus on to get authentic product in the market, as mentioned in the our Ayurvedic text or our herbal pharmacopoeias. So there are traditionally, if you can see most of, because all of many of us are from the pharmacognosy background here in this FDP. So you can see the five major categories: organoleptic, botanical evaluations, physical controls, physical parameters for quality control, chemical parameters for quality control, and biological parameters of quality control. So in biological, we also discuss toxicological parameters. And here in this presentation, we are going to see some new things which we generally encounter in quality control of our drug. So moving on to the next slide. So the major thing, we observe morphological identification. We feel the difficulty in morphological identification of the drugs. Like you can see in both the images, the yellow flowers you can see. So these flowers, if you look in the daytime period at 12 p.m., the first view, top view and side view, you can see. After 30 minutes, the flower starts to shrink, and after another half hour, or it means from 12 p.m. to 1 p.m., the flower changes its shape. So if these flowers are collected at different time, so and dried, they are dried as such. Then after drying, you can get three different forms of flower, morphological forms of flower, which is very difficult for a taxonomist to correctly identify. as per the pharmacopoeial matching of the standards. And similarly, you can see that another image. It is the same species with different look. So these things, these morphological differences in the drug or in the raw material which we get in the dried form, uh, they they make confusions in the mind of your taxonomist. Uh, it depend on the experience of taxonomist. If it is his or she is experienced, then they can easily identify. But in case of newcomers, it will become very difficult to identify the same species. So next slide, please, ma'am. There is another classical example of Rama Tulsi and Shama Tulsi. They both are two different species. One is Ocimum sanctum Rama and Ocimum tenuiflorium Shama Tulsi. Morphologically, if we see them, they both are same. But since because they are different species, they have different genotype, they have different characters. Even the content they have, like that of biomarker eugenol, the content might vary. So for actual or for factual authentication of drug material, to establish the identity of the drug material, we have to look for some more advanced features or advanced technologies for identification of actual identification or accurate identification of the plant products. So next slide, please, ma'am. So there is a term plant metabolomics. Most of us we have gone through metabolic studies and metabolites in the drug. Next slide, please. So if we see, there are different metabolites which are present in a herbal medicinal plant, be it a raw material, be it a semi-finished product, or be it a final finished product. So these metabolomes, they comprises only organic compounds with low molecular masses, which we call metabolites. And there are two types of metabolites we generally see in the drug, uh, uh, in our drug, like primary metabolites, which has got a wide distribution in the plant or in the living things, like carbohydrates, proteins, and all. And they have direct involvement in the essential life process of the plant. So on the other side, we have secondary metabolites, which have specific actions, which are species specific. Like we have seen, both the drug in the last slide, they are tulsi, but their content might vary because of the specific, because of the species variation. And if we see the distribution of secondary metabolites, it has got limited distribution in the living things, and they have a particular biological action. So plant metabolites, yeah, or the metabolomics studies of the drug compound is a very important parameter if we talk about the quality control or standards. So in the next slide, please. In the next slide, you can see a plant. We generally study in pharmacognosy that we use the leaves, we use the drugs, we use the roots or the fruits. So advanced techniques, due to the development of advanced techniques or evolvement of the advanced techniques, we are concerned with the leaf metabolomics alone. If we talk about the uh, if we talk about the leaves of vinca, the Sadabahar plant, so we have three different metabolomics, like metabolomics of the leaves, metabolomics of the stem, and metabolomics of the roots also. So at these three different plant part, we get different fingerprints of that particular plant. Same plant, but we have different fingerprints. So uh, in metabolomic studies, what we do, we generally prepare the samples, like you can see here. We prepare the sample by quenching in liquid nitrogen, and then the extract which we get of metabolite, it is then forwarded for a targeted or non-targeted analysis using the modern techniques, modern analytical techniques like 1-HNMR, LCMS, and gas chromatography mass spectroscopy. So on the basis of these data we obtain from those spectrophotometric analysis, we interpret that data, and after data processing we get uh, a print, like you can say in the right corner you can see the image. So these different blocks or different color, they shows the different concentration, presence and absence of the various secondary metabolites that are present in a given sample of drug. And after getting this, you can see this chromatogram or this fingerprint of a particular part of a particular plant, we interpolate, or you can overlay these metabolites on the known metabolic pathways or the known metabolic components which are supposed to present in that drug. We get an idea that what are the various components, different components which are present in a drug sample and which are absent in the drug sample. Even if standard chromatogram or the fingerprint of that particular drug, it might have more uh, it might have more or less number of these peaks or bands, but if we compare it with the standards, then it will give a clear idea about the presence and absence of some specific metabolites in the drug sample. So it is very easy method and quick method as compared to our traditional techniques of thin layer chromatography and PTLC and other techniques. So these are the need of time, which not only saves our time, but also uh, helps in overcoming the misidentification of the plant just because of geographical variations and other factors. So moving on to the next slide. This is like we have in the last slide, we have talked about targeted and non-targeted analysis of metabolomics. So this is an example of classical example of formulation, and we have developed its HPTLC fingerprint. And after developing its fingerprint, we obtain some RF values of various bands we have obtained, and we have calculated the RF values of those prominent bands in the sample. So on the basis of these different RF values that you can see in the picture above, so if we consider it, it is non-targeted. We are not targeting any biomarker. So we get a normal fingerprint with the number of spots and number of other values in a particular sample of drug in a particular solvent, like you can see in this. So this is non-targeted, or you can say it is global metabolomics for a formulation. If we move to the next slide, we can see here you can see a targeted metabolomic studies where we have targeted our test sample for confirming the presence of some definite biomarkers or phytoconstituent that might be present in the ingredient drugs of my formulation. So here you can see we have used two different concentrations of bacosides and piperine and withanolides. In the right side of upper image, you can see there are 6 spots of different concentration of 3 biomarkers which are present in a formulation which we are studying in the last slide. So you can, if you observe the presence, you can see the presence of these biomarker in the sample. So this give an idea. This is a targeted approach where we have a target to identify the presence or absence of a metabolite in a given sample of drug. So these are the I think HPTLC can be the indispensable article while uh, we are going to authenticate, or we are going to standardize, or we are going to quality control herbal formulation with respect to some biomarkers. So this is an example. This is on the basis of a simple technique HPTLC. Whereas in metabolomic studies, we also give consideration to other techniques like 1HNMR, GCMS, and other analytical tools that gives a clear idea about the presence or absence of some specific metabolites in the sample. So this is a new tool which most of us we are familiar with this. But if we club or we combine this technique with advanced techniques of HNMR and GCMS analysis and other techniques, we get more accuracy, or we get more accurate results, more accurate identification, more accurate quality control of our formulations and finished products. So moving on to the next slide. So where we can apply these techniques, we can use it for the species identification, like we have seen in the case of Tulsi. We can identify which is Oseum sanctum and which is Oseum tenuiflorium. We can also identify the habitat where the particular drug has been grown. Uh, a simple example I want to share that nowadays saffron is being grown in various parts of India and other countries. But if we talk about the natural habitat of saffron, it is in temperate climate of Kashmir and Ladakh. So if we grow these things in our labs or in our other areas, that will give a different pattern or different fingerprint of that particular plant, and that will help in differentiating between a saffron from the Ladakh or Kashmir region and that which is grown in the laboratory. So this technique can also be helpful in cultivation and the quality evaluation of our drug in the fields. It can also help in detection of heavy metals and agricultural pesticide residues in our drug sample. Also, these techniques can be helpful in identification of optimum growth age of our drug, and harvesting time. It can help in determining the appropriate harvesting time for our drug, at which the phytoconstituents or the biomarker for which we are using this drug is in optimum concentration. So it also helps in determining the optimum method for processing of drug. Like most of us, we know that after processing of herbs, mostly the phytochemicals they got a change in structure, and what fingerprint we are getting in raw material TLC, we are we feel difficulty in repeating or getting the same chromatogram in the processed product. So these techniques, advanced techniques, can also help us in designing an optimum processing technique, using which we can ensure the presence of optimum concentration of the phytochemicals in the drug or in the formulation. So another application of these techniques are in herbal marketing. We can identify the substitution and adulteration of the drug. So these techniques, they are generally being used for quality control. And right now, Chinese medicinal material, China is holding a large share in the herbal drug trade as compared to our country. We only have 0.5% share in the international herbal drug trade, whereas China is having around 21 or 22% of the share. So Chinese, they are doing these things. They are utilizing these techniques for quality control of their raw material and finished products. So that is why their material is authentic and their material is acceptable in the international level. So moving on to the next slide. These are examples of some materials which are analyzed for metabolomic studies. Achilea mellifolium, we use TLC, HPLC, and UV. So these are the example of various techniques which we can use for actual metabolomic studies of our drug. So moving on to the next slide. So actually, we most of us we know that before using a raw material, we have to get it authenticated from a taxonomist. Uh, we face various difficulties, like in the case in the slide we have seen that Osmium sanctum and Osmium tenuiflorium, they morphologically resemble, and for a taxonomist it becomes very difficult to analyze the difference between the both the drugs. Even on the phytochemistry level, if we calculate the percentage, we get some mixed results. So to refine that problem which is associated with the proper identification or authentication of the plant, the scientists have developed some DNA-based techniques for herbal drug authentication. Even Delhi University Department of Botany, they are busy in a project, a government project for DNA for developing DNA fingerprints of the various herbal material or various plants which are being used in the country. So what we get here, please move on to the next slide. Traditionally, we used to prepare a herbarium for the drug, and we have to send it for taxonomists to analyze the plant material for its identity. So nowadays, we are using the genome, uh, genetic makeup of the plant for its identification. So, what is the basis of this? Uh, this process, the DNA-based fingerprinting, the genome of each and every plant is very unique and specific. We all know very well. And in the case of those drugs which are very closely resemble, which are very closely related morphologically and phytochemically, and they are generally very difficult to be distinguished from each other. These techniques, DNA fingerprinting, it becomes indispensable and trustworthy tool for the authentication of the drug. So DNA-based identification techniques, they can be applied for both herbal material and finished products. However, the task of distinguish between plant species is more challenging in comparison to the animals, as the gene diversity is more in plants as compared to animals. Moving on to the next slide. What this DNA fingerprinting uses. Actually, like in this slide, you can see chloroplast is there, a plant cell is there, and we have isolated this plastid. So in the plastid, we have a sequence of genome, like you can see in the image, a ring-shaped structure you are seeing in the right side of the image. So there are specific regions on that particular genome of the plastid. There are regions like METK, they are RBCL, they are specific identified regions which are present in each and every plant. So what we do, we map the genetic sequence in these particular areas, these particular regions. So after amplification, uh, we get a clear image of the various the barcode of a particular plant, and this sequence it can also vary from species to species, or even from the various different plant parts. So the DNA fragment, the DNA sequence fragment is considered as a molecular marker for species identification as far as we talk about the DNA fingerprinting of the drugs. And this standardization method can be achieved by creating a unified database. And if we look at international level, there are various libraries throughout the world, like the Barcode of Life data system. They have coded, they have mapped the genome sequence at particular regions of various drugs. We have consortium for Barcode of Life, CBOL, medicinal material DNA barcode database, MMDBD, international barcode of life projects, IBOL, and GenBank, which is developed by NCBI, traditional Chinese medicine database. So these are the libraries where the barcode sequence of a particular or various herbal material they have stored, and you just have to get your barcode DNA fingerprint of your test sample which you want to authenticate or which you want to cross check, even it is from the finished product or from the raw material, and you just have to match it with the library, with the sequences which are given in the library mentioned here in the slide. So a software generally do these kind of things. You just got a result which includes the matching percentage. So they mention in the report that 85% or 90% of this plant matches with the existing standard database. So there are some relaxation given for authentication of plant due to genetic variation or due to geographical variation, but no doubt this technique is very well can be applied for proper identification and authentication, and you can say authentication of the drug. So these ideal there are certain conditions for getting these barcodes at different regions. So the ideal barcode must be it should have rapid enough evolution and sufficient nucleotide sequence variation, which is capable of distinguishing between species, but also have some limited intra-specific variation. So the we use PCR techniques here in this technique. So moving on to the next slide. So there are given references of various regions and different barcoding techniques. Like initially, we used to focus on a single region on the genome of a particular drug, but because it is not reliable to rely on a single region for getting barcode, authentic barcode. So we migrated to the comparison of two different regions like ITS region, TRNS region, MATK region, RBCL region, and we simultaneously combine the variation in the genetic makeup or the barcodes of these particular regions. And on the basis of 2-3 areas, regions, comparison of 2-3 regions, we got the result or outcome for proper identification of our crude drug or the drug material or even the finished products. So right now we are using NGS, which is next generation sequencing, along with the, or you can say coupled with high resolution melting analysis, which is used post PCR. And this technique is the latest technique which we use for the efficient identification or proper identification of the drug material. Moving on to the next slide. So there are some examples of different species along with their analyzed genome regions, which gives clear idea about the identity of that particular drug. Like if we look at the first Angelica species, here we analyze the different regions like METK, ITS, ITS2, RBCL, PSBA, and TRNH. So what these studies or these region genomic sequence gives an idea about, the DNA barcoding is used to identify the different, like here we have example of ITS region. This region gives a difference in the barcode to identify between Angelica anomala and Angelica daurica species. So the closely resembling species, morphologically indistinguishable species, can be distinguished or can be properly identified using these DNA barcoding techniques. So there are other examples. In Fabaceae family, ITS2 region, which is present on the plastids genome, it gives an efficient, powerful marker and potential barcode to distinguish between the various species of Fabaceae family. So using the barcodes or comparing the barcodes of distinct or specific regions on the genome of plastid, we get an idea about the actual identity of the drug. So these are the latest things which are being used by the international botanists and taxonomists for proper identification of the drug. I think most few of us we might know these techniques, but yes, most of us we are unaware of these techniques which are being used, and even we do not have such facilities for this genomic mapping in easily available in our country. So this is that demand of time that we shift toward the most sophisticated techniques for herbal drug identification authentication. Why I am focusing on the authentication and identification of the drug? Because most of our finished product which are being consumed by our citizens in India, they are generally they are not standard products. They are not standard raw materials which we are using. So moving on to the next slide. This is the focus area. Intrinsic toxicity evaluation. Like in the traditional way, we use various toxicological parameters like heavy metal analysis, aflatoxin analysis, analysis of pesticidal residue in the drug. So here, what we do, we generally focus on the presence of some phytochemicals because of which the toxicity occurs in herbal plants. So complications are more when we have a mixture of drug, or generally Ayurvedic formulation, they are a mixture of drug, or even herbal formulations. We generally use complex mixture of herbs so as to get therapeutic efficacy. Anyhow, so the complications are more with the heterogeneous complexes, where the effects they are unpredictable. So there are examples of some toxic chemicals which are formed in the plant, and they possesses toxicological properties. And if we consume these product for a longer period of time, we get some toxicological effects in our body, like pyrrolizidine alkaloids. They are hepatotoxic, genotoxic, cytotoxic, and phototoxic. We have got furan derivatives, epoxy diterpenoids, alkyl benzenes, and zincholic acids. So these they are embryotoxic, they are cytotoxic, they are neurotoxic. So presence of these chemical constituent must be limited in within ranges, within safe limit in our drug sample. And generally, have a specific we do not have a specific guidance or targeted approach for identifying these chemical constituent in the drug samples. Moving on to the next slide. Here we generally use the acute toxicity studies for herbal drugs. We perform acute oral toxicity using various guidelines like OECD 420, which is a fixed dose method, OECD 423, which is the acute toxic class method, up and down procedure in OECD 425. These are the various guidelines which are being mentioned in OECD guidelines for performing oral acute toxicity in the lab animals. For acute dermal toxicity, we go for the fixed dose method, OECD 402, which is a fixed dose method. And for inhalation toxicity, determining inhalation toxicity, which is generally performed for screening essential oils, we use the OECD 433 and 436 guidelines. So there are various there are mentioned procedures in under these different mentioned procedure under these guidelines, which uh, one have to go with for screening their product for minimizing or for analyzing the toxicity present in their formulation or in their raw material. So this is the traditional method we used to follow. Here we have some limitations. Moving on to the next slide. Uh, what we get generally, we have seen in CPCA meetings, when we put forth our proposal for studies, in vivo studies, there arises very ethical issues, and there is a general tendency of the members of CPCA CA members. They reduces the number of minimum number of experimental animals, and we cannot have a sample enough sample of animals, or you can say the enough number of animals to claim the activity of our drug. So generally, we then follow some biostatistical analysis, and we increase the number for getting favorable results. So this is the practice we generally follow in animal studies of herbal drugs. So technological processes progresses, they have led to the development of some in vitro models. There are some in vitro models that we can use for in place of in vivo studies, and they are superior in mimicking the in vivo conditions than the original ones. So we have 2D cell culture techniques, we have 3D cell culture techniques, we have organ on chip, which we are going to be used for evaluating the toxicity, intrinsic toxicity of the drug. So these new models, they have exponentially evolved, and but if we see they have limitations also, but because they do not mimic the accurately the organs physiology, so they have some limitations also. But no doubt, if we compare it with the animal studies in vivo which we perform on laboratory animals, for which we have a limited number of animals, so these techniques, they are advantageous. So the limitations we have in 2D cell culture is the altered cell morphology, modified division processes, and lack of interaction between the cell and the extracellular environment, and homogeneous distribution of the resources, and unlimited access. Like in 3D, we overcome some limitations of 2D cell culture, and in 3D we get a tissue model, 3D printed tissue model, and which have improved prediction of bioactivity. Moving on to the next slide. Here you can see the advantages and disadvantages of 3D and 2D cell culture. What we use here, suppose we have to perform hepatotoxicity studies. So what we do, we isolate hepatic cells from animals, or even we can also go for human cells, and we culture them in the laboratory. We divide them using various in vitro nutrient support. We get a 2D culture, and likewise we also get 3D tissue printing, also 3D tissue printing and 3D tissue culture. And instead of using organs, instead of using animal organs, instead of using the whole animal, we generally perform our studies on these isolated tissues. In vitro, we study these things. So if we compare with the animal studies, or with the various metabolic pathways in the living organisms like laboratory animals, here we get a direct result, a direct application of the medicament or the drug on the tissue, where we are going to evaluate the toxicity, or we are going to screen the toxicity of the drug. Even we can use these models for evaluating the therapeutic efficacy of the drug on the particular organs. So we have 2D culture, we have 3D culture, we have organ on chip, and again we have body on chip. They have some advantages and disadvantages. Like I have already discussed the advantage and disadvantages of 2D and limitations of 2D cell culture techniques. So as compared to 2D, 3D technique, they have improved resistance to drug-induced cell death. They have improved drug metabolism by increasing the expression of enzymes, which is generally absent in 2D cell culture techniques. So moving on to the next slide. Uh, there are recent advances, like you can see in the first image, you can see organ on chip. This chip contains the human airway model. So its name is Epi Airway. It is a registered product or patented product from a company. So what they are doing, they are using the your lung tissues, and they are mimicking the activity or physiology of the lungs on these chips. And they have also supplied with the blood supply and other things, nutrition support on these chips only. We have to generally place our product in the wells which are given on this chip, and we get the results. So for testing the inhalation toxicity of any drug, we can use these chips, and this is a patented product, or we can say it's a research product. Another example you can see is a vascularized 3D printed tissues. Like in 2D, we generally won't have the blood vessel supply in 2D cell cultures, but in 3D printed tissues, we are able to provide vessels, blood vessels in the tissue, and this is a classical example of epi skin. The second image of vascular 3D printed tissue, uh, it's a it's a print of skin tissues. So it's a reconstituted human epidermis, and it can be used as an alternative in analyzing the toxicity over your skin. Or for dermal toxicity studies, we can use these epi skin. So these techniques, they have potential to rationalize the field of revolutionize the field of for medicinal product safety evaluation. But since the techniques of DNA barcoding, the US, German, and Europe, they are using these techniques from last 30, 40 years, but still they are not available in our country. And if we talk about these organ on chip and 3D printed tissues, it will take another 2 decades, as I think, for us to use these techniques. It's a transition phase. They are in research. Maybe they have some their different effects as compared to the normal behavior or normal therapeutic benefit effect which we get from the living tissues or in the intact body. Next slide, please. There are other models which we can use apart from the traditional models. We can use Zebrafish model, which is a highly used and appreciated toxicity screening tool. And it is generally simple and inexpensive as compared to the animal models. Adult zebrafish can be used, but their embryos can also be used for embryogenic studies, where we have to study the effect of drug on embryos, or simply to analyze the embryo toxicity. We can use these zebrafish models. The advantage of this model is the transparency of embryo. You can directly observe the changes which are undergoing in the embryos directly from your eyes. So these models facilitate the toxicity examinations during different developmental stage, and the visualization of organogenesis is possible using some dyes and transgenic strains of zebrafish. So various drugs that have been studied for their toxicity using the zebra fish model, like Hypericum lanceolatum, Piper sarmentum, grape example, Hypericum lanceolatum, Oren orbiter. There are various other drugs also which have been studied for their toxicity using these models. So moving on to the next slide. Next slide, please. So till now we are concerned with the toxicity which we can observe. There are certain toxicity like the toxicity of gene and the toxicity to the fetus, the carcinogenicity. These are the toxicity we generally do not consider while evaluating the toxicity parameters traditionally. So if the toxicity occurred at genetic level, it has to be studied well, because there are certain drugs which are belonging to the families Borginaceae, Astraceae, Fabaceae families, and the genus of Symphytum and Proletaria, Heliotropium. They biosynthesizes by pyrrolidinone alkaloids. These alkaloids that are having genotoxicity, cytotoxicity activities, they are very, very much dangerous if taken in large doses. So there are examples of various phytochemicals like SRONs, SRaldehydes that are very toxic taken in for a long period of time. So what we are focusing, we have to screen our drug if we want to prepare our formulation for international standard. We must have a formulation which do not cause genotoxicity or carcinogenicity or various other type of toxicity. So we have various methods for determining these toxicities, like the FDA's and the ICH. They have prescribed a whole battery of you can say the test parameters. They are not relying on a single test for determining the genotoxicity or the mutagenicity or your carcinogenicity. They have issued guidelines. They have issued uh, a, a series of tests to determine the genotoxicity and carcinogenicity of the drug. Like you can see, for determining the mutagenicity, one can go for MAs, which is a bacterial reverse mutation test. Uh, for mutagenicity, we can also go for mouse lymphoma assay, in vitro mammalian cell genome mutation test. So these tests can be used for mutagenicity. For screening a drug for primary DNA damage, we can go for COMET assay. For chromosomal damage, to detect the damage in structural and numerical aberrations, we can go for chromosome aberration test, micronucleus test, LMCPA test. So these tests, they are prescribed by FDA and ICH for screening the phytopharmaceuticals or your herbal ingredient, herbal formulations for genotoxicity and carcinogenicity. Your your formulation must not have any potential or any chemical constituent in a toxic dose. So for this, moving on to the next slide. Next slide, please. Okay, so there are omics-based toxicological studies. Omics, which is a Latin term, which means totality. It relies on comprehensive integrated evaluations. So what we do here, this technique is based on the reflection of interactions between the toxic xenobiotics, yeah, toxic chemicals, and the biological systems at cellular level. So here we are observing the interaction between the tissues, between the biological systems, with the toxic chemicals or xenobiotics. We can simply say it. So for these studies, we have, if we do the study at transcription level, if we study variation, if we study the effect of the genotypes on xenobiotics on the gene transcription, it is called toxicotranscriptomics. In toxicoproteomics, we screen the effect of your toxic chemicals on the protein alterations as a consequence of exposure to the xenobiotics. In case of toxicometabolomics, we study alterations in the homeostasis and metabolic control of the organisms secondary to the exposure to a toxic compound. So these are the new techniques, omics-based techniques, which we can utilize uh, for determining or for screening the our drug sample for the toxicity, genotoxicity, or carcinogenicity of the drug. Moving on to the next slide. So I have included this slide to focus on the parameters which we currently use for quality control as a quality control parameter in our studies. So this is the last slide. These toxic these parameters, they are generally we generally carry out these screening. We generally screen our drug sample for these toxicity like microbial contamination. We use agar plate, colony counter method. We perform specific tests for specific microorganisms. We perform mycotoxin estimation using TLC, LC, or GC. Even immunological method like ELISA, they are also used. We have pesticidal and insecticidal residue determination using uh, liquid chromatography, gas chromatography, or immunological assay. Then we determine the radioactive contaminants in our species. We use gamma ray spectrometry, alpha particle spectrometry, and other techniques. For residual solvent, this is uh, uh, one of the point of interest for those who are going to study their extracts on the animals. We must ensure that our extract do not contain any traces of solvents before performing the test, because if solvents are there, they have their own toxicity on the animal. Even they have their own activity, and they can interfere. The results cannot be well interpreted. So for that residual analysis, we perform mass spectroscopic determination, gas chromatography we can go for. For toxic heavy metal analysis, we go for atomic absorption spectroscopy, and this is a very primitive technique which we are using for heavy metal analysis, atomic absorption spectroscopy. And right now, nowadays, we have inductively coupled plasma mass spectroscopy for determination of heavy metals contained in our drug sample. So, being pharmacognosy graduates, postgraduates, and doctorates, we must have to ensure the safety. We have to ensure the identity, proper identity, authenticity, safety, and efficacy of our herbal formulations. Only then we can expect that our share in the international market it can be compared with other countries. So this is very unfortunate for all of us that we generally neglect all these things to whatever the reason will be. It might be the lack of time, it might be the interest in money, or in the lack of resources. So the reason can be anything, but these are the factors that we must have to taken into account while performing the quality control and standardization evaluations of our drug. So it is better we can go for new techniques which are available in the uh, international level. So thank you so much. This is all from my side. If you have any query, because this is one-way communication, I think. So you can also mail your query to my email ID, which is rahulkcsji@gmail.com. Thank you so much. Thank you.
Thank you so much, sir. Now participants can post their queries in comment section, sir. Please see once.
Thank you, ma'am.
Sir, there is a doubt now being asked by one of the participants. Can you please explain the
Okay.
scope of metabolism in the in the drug development?
Uh, Very good question. Uh, as I have already expressed that we have to ensure the presence of each and every phytoconstant in our formulation or in our drug product. So metabolomics means the study, or you can say screening of each and every phytoconstant. So the if we have a presence of some toxic compounds, it must be taken into account because it might interfere in the drug development. Or if we have some biomarker like in turmeric we have curcumin, until and unless we know the exact percentage of curcumin in turmeric, or uh, uh, in various varieties of turmeric, if we know, until and unless we have the concentration, or we have the uh, uh, clear idea about its presence, we can not go for drug development. We have to ensure, because in drug development we have to specify the exact concentration of each and every constituent. So for that we must ensure that each and every constituent which are present, we should ensure their percentage in the sample. We should ensure their safety profile in the sample, like MSDS we get in synthetic drugs. So these are the things which must be taken into consideration while talking of drug development in herbal drug. So I have another question from Mr. Krishna Kumar Keti. Uh, does we use any tool like AI in comparing metabolite spectral data to reference databases? So right now, I don't know whether we have such kind of AI tool, but in future, it is it might be possible that we can have a tool like that. But again, we have some limitations like genetic variation, geographical variations. We can have because in herbal drugs, there is a term biotransformation. It takes place. So we have different concentration of phytoconstant at different times of storage also. So it is very difficult for herbal drug to get the application of AI, but nothing is impossible. Surely in this decade we can have data. Okay. Another question is, does metabolomics sound relate to primary morphological character, or may also be secondary morphological character during metabolic study? Uh, no, metabolomics is all about all the metabolites, be it primary or secondary, present in the drug. As far as the morphological characters they are considered, uh, we can we can assume that these techniques can be used to uh, to differentiate those species which are very difficult to be identified morphologically. Is there any question? Any more question? Any query? Okay, next question is from Mr. Kulvin. How does metabolomics detect any adulteration in herbal product? Like I have shown you two slides of targeted and untargeted, non-targeted approach. So if you want to detect any adulteration in the product, you will get a different chromatogram of a different fingerprint, a different chemical fingerprint of your product, and comparing the fingerprint of the standard drug along with the herbal product which is adulterated, you get the comparative results can easily give an idea about adulteration in the sample. We can detect adulteration in herbal product using metabolomics, because adulterants, they will give you, definitely they will give you their own bands in the chromatogram.
Thank you, sir. No more
Thank you.
queries, sir. It's
Thank you
my
so much.
pleasure to take today's session, sir. I sincerely thank our
Thank you.
resource person, Dr. Kaushik, sir, for delivering extraordinary presentation regarding current trends in herbal drug research, sir. Thank you so much once again.
Thank
And
you.
I would like to
Thank you so
thank
much.
our organization for their continuous support and trust in us for conducting FDPs. And I would like to thank our principal, sir, Dr. K. Achyut Kumar, sir, for guiding and support. And also, I would like to thank our faculty members and research scholars who have been participated in today's sessions. Thank you.
Thank
Thank
you
you
so
all.
much.